In today's PowerPoint, we're going to talk about the control of the cell cycle, and the way the cell cycle is controlled is through cell checkpoints. So control of the cell cycle, the checkpoint system is the cell cycle is controlled by stop and go chemical signals at critical points throughout the interphase, mitosis phase, and cytokinesis in the process of the cell cycle. The signals indicate if the key cellular processes have been completed correctly, and if they've been completed correctly, they can go on to the next stage. If they have not been completed correctly, they will signal a stop and prevent the cell cycle from continuing. The checkpoint overview, there are three major key points. The three key points are the G1 checkpoint, the G2 checkpoint, and the M or mitosis checkpoint. So at G1 checkpoint, the question is, can DNA synthesis begin? So has everything that is needed to be completed in G1, so the growth phase and gap phase, has everything happened that needs to be completed in order for the DNA to be replicated? In the G2 checkpoint, the question is, has DNA synthesis been completed correctly, and is the cell ready to commit to mitosis? So if the DNA has been replicated correctly, it is ready to go into the mitosis phase. The M checkpoint is the spindle checkpoint, and the question asked is, are all chromosomes attached to the spindle? And can sister chromatids separate correctly? So we know that the chromosomes have to eventually line up on the metaphase plate, so the spindle fibers need to attach correctly in order for the sister chromatids to be pulled apart and become individual chromosomes for each daughter cell. The G1 checkpoint is the most critical checkpoint. It checks for the cell size and the genetic material. If the cell receives the go signal, it proceeds in the cell cycle. The internal signals are cell growth, the cell nutrients, and the condition of the DNA. The external signals are the growth factors. So if cell does not receive a signal, it exits the cell cycle and switches to G0 phase. The G0 phase means that the cell is not going to undergo the cell cycle. It's going to be stuck in the current state that is in. It will not be replicated. So it won't divide and it'll stay in its form. So in G0, the cell is non-dividing and is in a working state. In the G2 checkpoint, the check fors are the cell size and the nutrients again. It's also looking to see if the DNA has been copied during S phase, and is the integrity of the DNA of a decent quality, so was it copied correctly? If the cell receives the go signal again, it proceeds with nuclear division or mitosis. If the cell does not receive a signal, the cell will be destroyed. It will undergo something called apoptosis, which is programmed cell death from your normal cell. And if the cell signal is not received and the cell is not ready for, or not completed correctly with the DNA being copied, the cell will have programmed cell death and die. In the M checkpoint, this is during metaphase. So the M checkpoint is mitosis, but specifically metaphase. So we know PMAT. We know that we have prophase, metaphase, anaphase, telophase, followed by cytokinesis. And in metaphase, we are checking the spindle fibers of spindle fiber attachment. We're making sure that the sister chromatids can separate properly so that the daughter cells can get a complete copy of the DNA. If the cell receives the go signal, it divides. If the cell does not receive the signal, it will undergo the same cycle, programmed cell death, which is apoptosis again. So you have your centromere in between the attaching two sister chromatids, and you have the spindle fibers attaching to that, and they will line up around the metaphase plate when the spindle fibers attach to allow for the chromosomes to be pulled to the opposite sides. So at all of these go signals, the go signals are called, there's an enzyme called cyclin. So cyclin is an enzyme that will build up, and that is the go signal for all of these go phases. External signals, we're looking at the growth factors, really the coordination between cells. There are protein signals released by the cell bodies that stimulate other cells to divide. There's density-dependent inhibition. This is where the crowded cells will stop dividing. So when there are enough cells in a certain area, no more cells will be divided or produced. So there will be no more cell division. So each cell binds a bit of growth factor. So not enough activator left so there isn't enough activator to trigger division in any one cell. Anchorage dependency. To divide, cells must be attached to a substrate. So they must be physically, so it's a touch receptor sort of dependency, that they have to be anchored at a certain point in order for them to divide. Mitosis in cancer. So this is when new cells go terribly wrong. So we have these stop and go checkpoints that allow for our cells to grow properly, but sometimes there can be a little haywire, and this can lead to cancer. So when is mitosis a good thing? So when do you have to add and replace cells? So when do we need cell division? We need it for growth and development. So when we're a fertilized egg and we develop into an embryo, fetus, baby, adult, as well as repair. So if you scratch yourself, your body will replace the skin cells where you scraped yourself. It'll start off as a scab as your skin regenerates to fill in the spot where you have scraped yourself. We also have replacement, limit cell size, and for asexual reproduction. When is mitosis a bad thing? Mitosis is a bad thing when cells reproduce and they are not needed. These cells that grow can take over organs and don't do the right job. They just make copies, and copies and copies. There isn't a stop and go, so they don't have the red light, green light system going on. When they're continually growing, this could potentially lead to cancer, and cancer can potentially lead to damages in the organs. So in this picture to the right, you have cells. So in the picture to the right, you have a mammary gland, and there's an overgrowth of the tissue cells, and that can lead to a tumor. That tumor can be benign or malignant, but I will talk about that in the coming slides. So why would cells just make copies? So if DNA gets damaged, cells stop listening to the correct instructions. So typically we undergo apoptosis, which is the programmed cell death, but if cells stop listening to the programmed cell death, the cell can keep dividing and dividing with the damaged DNA. So These errors are called mutations, and we've talked about mutations in the past. Specifically, if it stops the listening stops if there are mutations and the cell ignores the cell checkpoint, so it's ignoring the stop and goes. The causes for these mutations can be UV radiation, chemical exposure, radiation exposure, heat, cigarette smoke, pollution, age, and genetics. So over time, naturally with your age and your genetic disposition, your body and your cells will undergo mutations. They aren't necessarily bad, but when you're exposed to different carcinogens are any environmental exposure you have that could potentially mutate your cells and potentially lead to cancer. So one of these specifically won't just lead to cancer, you have to have multiple mutations over time. So that's why with age, as you age, your body's going to undergo and be exposed, or is going to undergo multiple mutations, increasing your risk of cancer as you get older. Okay, so growth factors in cancer. Growth factors can create cancer. So proto-oncogenes are a normal type of cell. It normally activates cell division. It's part of the growth factor genes, and when it becomes an oncogene, which is the cancer causing, so it has mutation, it can lead to cancer. So proto-oncogenes are typically off, but if the oncogene or proto-oncogene is turned on, it will become this oncogene, which can cause cancer. An example is RAS. RAS is an enzyme that activates cyclin, and cyclin is an enzyme that is involved in the cell cycle stop and go process. So it's part of what we were talking about a little bit earlier, the checkpoints and control. Another growth factor is the tumor suppressor gene, which equals the stop. So normally it inhibits cell division, so it limits the cell division. If it's switched off, this can cause cancer. The tumor suppressor gene is preventing these cells from rapidly growing, and if the tumor suppressor gene is turned off, that means the cells are just going to rapidly grow and potentially form a tumor. Specifically, you look at the protein P53. P53 is involved in repair of DNA as well as apoptosis. So if it is not present and it's not inhibiting cell division, you may have a rapid growth of cells that can lead to tumor. So development of cancer. Cancer develops only after an experience of 6 key mutations, also known as hits. The hits include unlimited cell growth, so turning on the growth promoter factor gene, the ignoring of checkpoints, so turning off tumor suppressor genes, so for example the P53 protein. The cells escape apoptosis, so it turns off the suicide gene, so cells will not automatically divide or automatically undergo programmed cell death. If there's a mutation in cell division, so cells become immortal and have unlimited cell division. So you turn on chromosome maintenance genes, the cells just rapidly divide. Also promotes blood vessel growth, so turns on blood vessel growth genes. And also if cells overcome the anchorage and density dependency factors. So you turn off the sensory gene, so cells are no longer paying attention to density dependency and will just rapidly overgrow, or anchorage, they don't need to be attached to anything to continue to grow. So pretty much cells are out of control and doing whatever they feel like and rapidly growing. So tumors I mentioned before, there's something called a benign tumor. This is abnormal cells at an original site such as a lump. So in this case, you're looking at a mammary gland. Benign is a good thing. Most benign tumors do not cause serious problems and can be removed by surgery. So if it's a benign tumor, that just means the cells just kept growing but haven't led to any cancerous problematic issues and are concentrated in one area. So if a tumor is known as being malignant, the cells have left the original site. So if a tumor is malignant, it will leave the original site, so it will metastasize. It can be carried by the bloodstream to other tissues. So if it leaves its original site, so if it leaves just the mammary gland and enters into the lymphatic system and spreads to the other body, this can lead to bigger and worse issues because it'll start producing more tumors and can damage functions of organs throughout the body. So the treatment for cancer are chemotherapy. So treatments will kill rapidly dividing cells. So you have chemotherapy and radiation. Chemotherapy specifically is a poisonous drug that kills rapidly dividing cells, whereas radiation is a high energy beam that also kills rapidly dividing cells. That's why when people undergo chemotherapy and radiation, they will potentially lose their hair because chemotherapy and radiation do not kill just cancerous cells, they're killing any dividing cells in your body. So they're just trying to get rid of the bad cells, but in the process may potentially get rid of the good hair as well. So that's one of the side effects. If you have any questions, please post them on Schoology, and I will talk to you in class. Have a good day.